I want to tell you about how we're using games to reconstruct networks in the brain, what is called connectomics today. You all know genomics. I'll try to tell you a little bit about what connectomics means. I want to start with something that's really interesting, I hope, to all of us, which is our brains. This is just one picture of it, and maybe we can start with a comparison. Who of you thinks that you have more neurons in your brains than there are people on Earth? So who thinks more neurons than people? Okay, and by inversion, at least half the audience thinks it's the other way around. Well, it's pretty crass. It's 85 billion neurons, and as far as we know, it's not that many people. So the brain, and each of your brain, contains much more players than Earth hosts humans. That's only the beginning of the story. The second part to it is that each neuron, as far as we know today, directly talks to roughly 1,000 other neurons by synapses, as we call it. Now, I want to ask you again, who of you has 1,000 friends on Facebook? Okay, some of you do, most don't. So our brains are networks that each of it are much more complex than even the human network on Earth. That is what we are facing. And if we want to understand the brain, it's natural that we want to map those networks, because that's the way to understand it. So we have to find methods to map the neuronal networks that are that complex. And the good news is, first, the bad news is that it's very difficult. So for 100 years or so, we haven't been able to do it because neurons are very small. They can be as small as 50 nanometers at some parts. At the same time, they extend over hundreds of micrometers, if not millimeters. So that is a huge scale span there between 50 nanometers and like 10 million or so nanometers in scope. So the problem is neurons are small and large, and that makes it difficult to image them. The good news is that we've recently found methods to map these complex neurons, and this is just one of those. All of what you're seeing there in white is one single nerve cell, and it's even better than that. So there are many structures to it, and it's really complex, and this has been actually known for quite some while, that they are so complex, but it was difficult to map them. Now, the new methods we have now since a few years work like this. You take a small piece of brain and then you put it into an electron microscope, and we have a new microscope developed in Heidelberg which is automated fully, so it runs for weeks and weeks and takes many, which is thousands and thousands of very thin images, and then there's a knife cutting off the top of the block that you put into the electron microscope, and by that basically you get a three-dimensional volume image of a small piece of brain at the right resolution, at the resolution we need to map those networks. So this is really great. We have now a new microscope and we can get data like this. This is just an example of many of nerve fibers running through a small piece of, in this case, retina at the same time. And one of those neurons there is reconstructed in red. As you can see, it's coming out there. This is just a small piece of the neuronal networks that we're facing. So this is the methods we have now. This is pretty exciting to us. A different view is this one. Now we are flying along a neuron. So now we are flying along an axon into that piece of tissue, and you can get an idea of the complexity of what we're talking about. So all of the rest of the stuff you're seeing there is also neurons, and they're just very densely packed. And this is a 3D rendering of how we're flying there. So just keep in mind, this is really difficult to reconstruct. And it's so difficult that computers currently can't do it. And this is, I know, a pretty daring statement to make in an audience like this, but you know, many settings where computers today cannot do what humans are able to do. For instance, visual scene segmentation, at least in noisy settings, is not solved algorithmically. And this is just one additional problem where all the algorithms we and all of our collaborators in the world tried are not good enough. So we have to use human annotators. And because we can't rely on a single annotator, we use many, and we've actually hired more than 300 students to do this reconstruction task by flying along the fibers in the brain. And what you get then is, again, this piece of data, but then as a next step, we can find all the cell bodies. That's reasonably simple, but then what is key is that in the next step, we can ask all of our students to reconstruct the network, the neuronal network in that piece of tissue, and this is what we have done with a small piece of mouse retina. We can fly into that a little bit at least, so you get an idea. So these are centerline reconstructions where people just flew along the cables without volume filling, and as you can see, there's still some space in there, and you'll see a more representative volume picture in a second. Okay, so this is what we can do already with the help of humans, and this is just to show it once. This is a matrix. This is a connectome, as we call it today. So you know genomics, which was mapping the DNA. We think we have now a new field in neuroscience where we are mapping connectivity matrices, the connections of nerve circuits or neuronal circuits, and the way to depict it is such a matrix where the strength of the connection is indicated by color. And we did this now for a small piece of mammalian brain, which is the retina in the mouse, and it's a big step forward. Now, what can we learn from this? There are many things we can learn from that in terms of circuits, but I also want to update your picture of your own brain. So you may have seen images like that many times, right? That is probably what you think a brain looks like neurons that are kind of connected to each other and fire some electrical signals. Now, this is not the right scale. What it really looks like is this, and this is an actual rendering from our data. It's much denser. This is, and we're lucky we're flying along a blood vessel, otherwise we couldn't see anything. It's packed with neurons and fibers everywhere. We're just flying backwards again. So this is a blood vessel, and therefore we can see what is around it. Without blood vessels, no chance to see what's happening in your brain. So brains are super dense, densely packed circuits. Really amazing. Okay, cool. That is what we have done, and this was basically a big step because in the last century, very famous researchers did a worm and got a Nobel Prize for that, to do a small worm network. We did the mouse now in Heidelberg together with Winfried Denk over the last 5 years or so, but now comes the ambition, and this is why I'm here talking about games. We now want to go to the cerebral cortex, which is the part of our brains where we actually do the interesting stuff, which is thinking and talking and all of these kinds of things. And also, even mice do the most interesting stuff in their neocortex. And now we want to map circuits in the neocortex. And the task is just really amazingly large because we need hundreds of thousands of work hours. What we did was 30,000 work hours for the retina, therefore we hired students, and that was doable. 100,000 or many hundred thousands of work hours is not easily doable. So how can we address this, and what is our plan there, and what are we doing there? What we're doing is we're trying to interest more people in our task. And we developed ways of putting our data into the browser. And this is just an excerpt here where you can see now it's basically like a 3D panning, and you're flying along one of those processes. There's a lot of stuff in there. It's actually partly difficult to detect, but humans are very good at that. And we've tested that now with many students mostly, that you can fly along one of those cables in a setting that reminds you a little bit maybe of a flight simulator, where you're basically trying to steer or navigate through the brain. So this is one of the attempts we're making, and it's really like a 3D spherical projection of the data running in the browser. We're using WebGL, which was quite some challenge on its own, but now it can get actually even cooler. So this is a version where we have a 3D surface rendering also running in the browser of our original data. This is not yet reconstructed, and the task here is to find the tunnels, and if you like color more, then you can get this version where we're trying to colorize the isosurfaces. This looks a little bit puzzling, but in a second you will maybe see a tunnel there coming up, and this is again a nerve fiber that we want to follow. It's one of the wires in the neocortex of a mouse. Again, along those cables and those tunnels as we depict them here is where the mouse does recognition tasks which no computer can do today. So these are the ideas. We have several more game ideas coming up. So this is a more casual game-like setting that we're running on the iPhone and iPad, where you can swipe through the data, and the task is to follow one of those processes, and playing around with colors and stuff like that to make it maybe a little bit more interesting. But really, the task here is to follow cables through the brain. And if we do that, we can get actually to something that is, I think, very exciting. So we need hundreds of thousands of work hours, but the goal would be to use our common brain power to actually, for the first time, crack what nature built as the most powerful computational device we know of. And again, be reminded, none of our algorithms today is as good as even a mouse's cortex in detecting, say, an apple or a tree or something like that. And the real goal here is, and I think that's just a very ambitious but also exciting task, that we may be able to use our crowdsourced brain power to crack that code and ask how nature implemented this extremely powerful computer. In a way, it's like, imagine you're an alien coming to the world, and the first thing you find is an Atari computer, and you, for the first time, open it up and try to understand how it works by looking at the circuits. And actually, it's better because we can even read the software by looking at the networks in the brain. So this is the endeavor we're up for, and this is why we're using brain games to get there, because we need a lot of manpower and brain power collectively. And this is our excitement that I try to share. These are the people involved, and I hope you get an idea of where we're going there. Thanks a lot for your attention.